Okay,
Oh,
so today we're quite pleased to have with us Thomas Hill. He has rescheduled from his original time, and so we're really pleased that he can join us now. The title of his talk is Loss of Electrical Beta to Delta Cell Gap Junctional Coupling in Pancreatic Islets Contributes to Impaired Hypoglycemia-Induced Glucagon Secretion in Type 1 Diabetes. A little bit about Tom. Originally, he's a Canadian. He obtained his master's in biomedical and molecular sciences research from King's College London. His research investigated the impact of short-chain fatty acids on the regulation of beta cell insulin secretion from pancreatic islets under the supervision of Dr. Shanta Prasad. He completed his PhD at King's and investigating the placental islet signaling access in health and gestational diabetes under supervision of Dr. Peter James and Dr. James Bowe. He was awarded the Novo Nordisk Oxford Postdoctoral Research Fellowship and joined the labs of professors Francis Ashcroft and Patrick Roseman at the University of Oxford in the UK, investigating the regulation of pancreatic islet delta cell function and their complex paracrine signaling networks in health and disease. And then he joined the lab of Doctors Benjamin Davies at the Wellcome Center for Human Genetics at the University of Oxford in the UK, where he expanded his expertise into generating transgenic gene knockout and knock-in mouse models using CRISPR-Cas9. Since then, since 2024, he's been working as a postdoctoral research associate with Dr. Roseman, investigating the dysregulation of islet alpha and delta cell function and their contribution to prevent potentially life-threatening severe low blood sugar levels, hypoglycemia in type 1 diabetes. I'll drop his recent paper in the chat for your reference, and feel free to drop questions in the chat during the talk, and Tom will take questions at the very end of the talk as well. Thanks so much for joining us, Tom. We're really excited to hear your talk.
oh, thank you. And thank you so much for the kind introduction. And we're incredibly grateful for this amazing opportunity to share our work with the type 1 diabetes and sugar science community. So can everyone hear me okay? Is that everything's okay?
Yes, all
Okay.
systems go.
So, and I know this is typically shared at the end of the talk, but wanted to start off by sharing the teams and faces behind this work and where we're based. And most of us are mostly between two sites at the Oxford Center for Diabetes, Endocrinology and Metabolism, and also at the Institute of Neuroscience and Physiology at the University of Gothenburg. But this work also includes collaborators near and far, including the University of Ulster, Copenhagen, Coimbra, and the India Institute of Science. And we all work very closely together. And most of us, if not all of us, work on pancreatic islets, their function and communication in health and disease. And as we know, these fascinating pancreatic islets, of course, play a crucial role in controlling blood glucose homeostasis in the body, which comprise the well-known beta cells that release the hormone insulin, which lower blood glucose levels. And these can be shown here by these cells in the white in my immunofluorescent image of a non-diabetic human islet. And then there are the alpha cells that secrete glucagon, which acts to elevate blood glucose, represented by the red cells. But there are also these rare delta cells represented by these cells in green, and these only constitute approximately 5 to 10% of the islet that release the hormone somatostatin, and they're very understudied. And these delta cells contain long filopodia that can span more than 20 microns in length that enable them to contact multiple and even distal cells in the islet. And these cells are constantly in communication with each other, forming intricate paracrine and autocrine networks of stimulatory and inhibitory feedback signals, with some of them shown here. And highlighting one of these feedback networks includes the release of somatostatin from the delta cells, which acts as a potent inhibitor for both beta and alpha cell activity and hormone release by binding to and activating the somatostatin receptors expressed on the cell surface. But in type 1 diabetes, the insulin-secreting beta cells are, of course, largely destroyed by an autoimmune-driven attack. And this not only highly disturbs normal intra-islet communication, but it also changes the architecture of the islet, which can be shown here to the right in my immunofluorescent image of an islet taken from a long-standing type 1 diabetic donor. And in these islets, and particularly in donors with long disease duration, the islet shape becomes dysmorphic, where they lose their defining border and mostly comprise of the alpha and delta cells, which aren't destroyed in the autoimmune process. And the prevalence of type 1 diabetes is increasing at an alarming rate worldwide, with cases expected to double by 2040. But type 1 diabetes is not just a problem of high blood glucose, it's also a problem of low blood glucose, as individuals with type 1 diabetes have an increased risk of hypoglycemia, being a common and serious complication of insulin therapy and limiting factor in glycemic management. Where continuous glucose monitoring data have shown that individuals with type 1 reportedly experience, on average, around 6 episodes of hypoglycemia per week, lasting longer than 15 minutes. And approximately 30 to 40% of individuals with type 1 diabetes experience approximately 1 to 2 episodes of severe hypoglycemia per year, requiring the assistance from another person to treat, resulting in seizures or coma. And the fear of severe hypoglycemia significantly affects the quality of life for individuals with type 1 diabetes, resulting in less stringent blood glucose control. And the alarming statistic is that severe hypoglycemia has been reported to account for as much as up to 10% of type 1 diabetic deaths, and is also a cause of dead in bed syndrome for individuals with type 1 diabetes, particularly in children, as a result of severe hypoglycemia occurring during the night. And this increased risk of hypoglycemia and severe hypoglycemia in type 1 diabetes is attributed to the physiological defense mechanisms that act to prevent hypoglycemia in the body being compromised, which includes a loss of the chief counterregulatory hormone glucagon from the islet alpha cells, which is hugely problematic since glucagon is crucial for stimulating hepatic glucose production and raising blood glucose levels again in the body. And this was originally demonstrated more than 50 years ago by Gerich and colleagues, which can be nicely represented here in these graphs to the right, where we have individuals with type 1 diabetes in red and non-diabetic individuals in black. And in response to exogenous insulin administration at time 0, there is a normal increase in glucagon secretion in response to a fall in plasma glucose in non-diabetic individuals, but this response is severely blunted in individuals with type 1 diabetes. And why this impairment in glucagon secretion occurs is not fully understood, but potential hypotheses that have been put forward explaining this defect include, but are not limited to, a loss of islet autonomic innervation in signaling, a loss of intra-islet insulin and paracrine signaling from the beta cells, and also include intrinsic defects to the alpha cells themselves, where defects in alpha cell glucose sensing and metabolism, ion channel activity and electrical activity, which it was shown by Theo a couple of days ago, impaired autocrine glutamate receptor signaling, and also a loss or altered alpha cell identity have all been proposed to play a role. Now, the latter two hypotheses, of course, suggest that this phenomenon is caused by also caused by intrinsic defects that resides within the islets themselves. But what's also interesting has been routinely and consistently reported in clinical studies is that the presence of C-peptide and residual C-peptide found even in individuals who have had type 1 diabetes for 30 to 50 years are associated with a lower risk of hypoglycemic and severe hypoglycemic episodes, suggesting that the presence of the beta cells and beta cell function seems to be important and has a protective effect in reducing the risk of hypoglycemia in individuals with type 1 diabetes, and to keep this in mind for later. So we also wanted to take a look at this and to help answer the following questions as to what causes defective counterregulatory glucagon secretion from autoimmune diabetic islets in response to hypoglycemia. Does this defect also occur in isolated human type 1 diabetic islets? And importantly, can we improve this impaired glucagon response from autoimmune diabetic islets? So to investigate this, and with human type 1 diabetic donor islets for research being limited, we first used the spontaneous autoimmune non-obese diabetic mouse, or more commonly referred to as the NOD mouse, which is a widely used polygenic model for studying type 1 diabetes, which develops autoimmune diabetes with age. And in the study, we used young non-diabetic mice of around less than 7 weeks of age, adult diabetic mice, which were more than 12 weeks of age with a blood glucose concentration of 25 millimolar or higher. And we also used their age-matched non-diabetic littermates to represent adult pre-diabetic controls. Where the islets from both adult diabetic and pre-diabetic control mice show signs of immune cell presence, as shown here, or immune cell infiltration in the case of the diabetic islets, which are shown in these H&E stained images of islet sections taken at the disease states, where the immune cells are identified by these small nucleated cells surrounding the islet. But where the diabetic islets, of course, show a marked loss in the number of insulin-positive beta cells per islet when compared to adult pre-diabetic islets and young non-diabetic control islets, as shown in these immunofluorescent images, where the beta cells are represented by the insulin staining in white. And the neat thing about using the diabetic NOD mouse is that it models the problem of hypoglycemia in type 1 diabetes rather well, where if you take fasted adult diabetics, shown here to the right, and normal glycemic control mice, shown to the left in black, and you inject these mice with exogenous insulin by performing IP insulin tolerance test to mimic therapeutic hyperinsulinemia, where the blood glucose concentration is shown in the above panels, and insulin is injected in time 0, but additionally also at 30 minutes in the diabetic mice to really push their blood glucose down to a hypoglycemic range, then what we see is, unlike the control mice, which show a threefold increase in glucagon secretion in response to insulin-induced hypoglycemia, that this response is abolished from the diabetic NOD mice. And we can see this in the impaired glucagon response using the in situ perfused pancreas, which is a technique where we place an inflow cannula at the celiac artery that is connected to Krebs' physiological buffer, supplemented with our glucose concentration of interest, and an outflow cannula at the hepatic portal vein, shown here. The pancreas is then perfused with a buffer using a peristaltic pump, and the effluent is collected from the portal vein and subjected to hormone quantification. And this is a great technique that really allows us to keep the islet microenvironment, vasculature, and paracrine signaling intact. And what this revealed was, despite there being a stimulated increase in glucagon secretion when going from high to low glucose from the young non-diabetic pancreas, shown in black, and to a lesser extent in the adult pre-diabetic pancreas in gray, that this response was completely blunted from the diabetic pancreas, shown in red. And we also saw a similar pattern of glucagon secretion in the isolated islets from these mice at the different disease states, subjected to 1 hour static secretion experiments, where islet innervation is of course severed and the islets are removed from any extrinsic in vivo factors, where at low 1 millimolar glucose, glucagon secretion was highest from young non-diabetic islets, again in black, but lowest from adult diabetic islets in red, supporting this impaired glucagon response to hypoglycemia to be an intrinsic islet defect. So we asked, what causes this impaired glucagon response to hypoglycemia? Well, it's not due to alpha cell degranulation or exhaustion, as electron microscopy reveals that the alpha cells from diabetic NOD islets, shown here, are still granulated when compared to alpha cells from non-diabetic islets. And it's also not due to their exocytotic machinery being defective, as diabetic alpha cells still secrete glucagon in response to other stimuli, such as amino acids, as shown here in this perfused pancreas experiment, where the administration of the amino acid arginine at 5 minutes still leads to a robust stimulation of glucagon release from the diabetic NOD pancreas. And this has also been reported in individuals with type 1 diabetes who likewise show a glucagon response to oral and IV administered amino acids. And so therefore, this defect in glucagon counterregulation in type 1 diabetes is not due to an impairment in the alpha cell exocytotic machinery or their ability to secrete glucagon. Now, previously, it's been suggested that this impaired glucagon response to hypoglycemia in type 1 might be caused by an elevation in alpha cell paracrine suppression due to an increase in SMASM release from the neighboring delta cells, which, bringing back my fluorescent image of a human type 1 diabetic islet, where the delta cells are represented in green, and you can see they're in close contact with the alpha cells in red, where this proximity to the delta cells and their filopodia may even be higher due to the significant loss of the beta cells. And where SMASM release from the delta cells, as mentioned, acts as a powerful inhibitor for alpha cell glucagon secretion by binding to and activating the SMASM receptors expressed on the cell surface. So we also measured SMASM release from the same islet stat secretion experiment shown earlier, which revealed basal SMASM secretion at 1 millimolar glucose to be significantly higher from the diabetic islets, again shown in red, when compared to the young non-diabetic controls in black, suggesting that the diabetic alpha cells at low glucose are indeed subjected to elevated paracrine suppression due to an increase in SMASM release from the delta cells. So we next asked, well, are these diabetic delta cells more active then, since they secrete more somatostatin? So to investigate this, I performed live delta cell calcium imaging within intact islets that were isolated from a novel version of the NOD mouse, which I've generated here at Oxford, called the NOD delta GCAM6 mouse, which expresses an intracellular calcium sensor specifically in the delta cells. and showing representative calcium traces from these experiments with calcium activity from non-diabetic control delta, from a non-diabetic control delta cell shown to the left in black, and a diabetic delta cell shown to the right in red, where an increase in the number of calcium spikes in peak amplitude is reflective of an increase in calcium activity, that in the non-diabetic control delta cells, intracellular calcium activity was normally increased at stimulatory 10 millimolar glucose and suppressed at low 1 millimolar glucose. But in the diabetic delta cells, intracellular calcium activity failed to be suppressed at 1 millimolar glucose, where the spike frequency and peak amplitude was actually fairly similar to that produced at high glucose. So we then asked, does this pattern of secretion also occur in human type 1 diabetic islets? So we performed stat secretions on islets isolated from human non-diabetic and type 1 diabetic donors, which were roughly matched for age and BMI, which showed a normal stimulation in glucagon secretion from non-diabetic donor islets when going from 6 to 1 millimolar glucose, where each color corresponds to an individual donor. However, just as seen in the non-diabetic islets, hypoglycemia-induced glucagon secretion was significantly blunted from type 1 diabetic donor islets. But the type 1 diabetic islets also were shown to secrete significantly more somatostatin at 1 millimolar glucose. So we were wondering, well, what on earth is causing this increased somatostatin release from the islet delta cells at hypoglycemia? So we examined our non-islet secretion data a little closer, looking at hormone release levels conducted at different disease states, where we noticed that when plotting glucagon and somatostatin secretion against insulin content, that glucagon levels were reduced and somatostatin levels were elevated with a decreasing islet insulin content with an increase in disease progression, suggesting that the loss of the beta cells could be an underlying factor. So we wondered, well, why the loss of the beta cells? So the mechanisms controlling the regulation of somatostatin secretion from the delta cells is still an area of active ongoing research, investigation, but our lab and others have identified three main mechanisms so far. So the first is like the beta and the alpha cells, that the delta cells possess an intrinsic metabolic regulation that is mediated by the closure of the KTP channels, which triggers a membrane depolarization, opening of the voltage-gated calcium channels, leading to an influx of intracellular calcium that drives exocytosis of the somatostatin-containing granules. Now, the second is by extrinsic regulation from secreted factors, such as the incretin hormones GLP-1 and GIP from the gut, for example, or by intra-islet autocrine and paracrine regulation from secreted factors released from the delta cells themselves and surrounding beta and alpha cells, respectively. And the third is regulation from electrical coupling mediated by gap junctions to the neighboring beta cells. And this electrical coupling is largely mediated by the gap junctional protein connexin 36 that is expressed by both cell types that enable the transfer of ions. And this can be shown here in my immunofluorescent image, where we have two beta cells in pink at the top, which express connexin 36, particularly this beta cell. And you can just see squeezing in between these two beta cells is a delta cell phylopodia, which you can see in the merged image. Now, previously, the Roseman lab has shown that by stimulating an increase in beta cell activity in the physiological context of high glucose, that this depolarization and increased electrical activity from the beta cells can spread into the neighboring delta cells, also triggering a membrane depolarization and increased electrical activity in these cells via the gap junctions. But in type 1 diabetes, the beta cells are largely lost, and the delta cell expression of connexin 36, which is encoded by the gene GJD2, is downregulated in autoimmune adult NOD delta cells when compared to non-diabetic, non-autoimmune delta cells from C57BL6 islets when assessed by single-cell RNA-seq. And the same is also true for human type 1 diabetic delta cells when compared to non-diabetic delta cells taken from the published single-cell RNA-seq dataset. So we contemplated the possibility that, under non-diabetic conditions, that the high KTP channel activity and the hyperpolarizing electrical current from the beta cells also spreads into the neighboring delta cells via the gap junctions, keeping the delta cells electrically silent at low glucose. So we tested this concept using optogenetics, which uses a model of a non-autoimmune, non-diabetic C57BL6 mouse expressing a light-activated inward chloride pump called halorhodopsin, specifically in the beta cells. And this pump was tagged with a YFP, as shown here, which allowed us to identify the beta cells expressing the halorhodopsin. where if you shine 625 nanometer wavelength red light on the beta cells expressing this channel, then this will activate the chloride pump, causing an influx of chloride ions into the cell, triggering a membrane hyperpolarization. And we showed that this using patch clamp electrophysiology, where if you stimulate the beta cells with high 10 millimolar glucose, as shown here in this membrane potential recording of a beta cell exhibiting spontaneous action potentials, but then you activate the chloride pump on the beta cells by shining red light, that this hyperpolarizes and stops action potential firing, not just in these beta cells, but it also in the neighboring delta cells, as shown here to the right, which do not express the halorhodopsin chloride pump, confirming these cells to be functionally coupled. And these beautiful experiments were performed by Haiqiang and Caroline. And this was also the case at low 1 millimolar glucose, where light activation of the chloride pump on the beta cell, which is already hyperpolarized, also hyperpolarizes the neighboring delta cell by a further 10 to 12 millivolts, which can be strongly attenuated, however, when applying a gap junctional inhibitor called carbonexolone. So since the beta cells are electrically coupled to the delta cells, we then ask, well, what happens if you disrupt this electrical coupling, which inevitably occurs in type 1 diabetes as the beta cells are targeted for destruction? So to answer this, we tested the glucagon and SMASTAM response to hypoglycemia in static secretion experiments on islets isolated from young non-diabetic mice where the beta cells are still present and beta to delta cell coupling is intact, where we then treated these islets with and without the addition of a different and more specific gap junctional inhibitor for connexin 36 called mefloquine at 1 millimolar glucose. And intriguingly, what we found was that the addition of mefloquine increased basal SMASTAN secretion, which resulted in a suppression in glucagon release in a similar pattern of secretion to what was observed in the diabetic NOD islets. And the application of mefloquine also triggered a similar secretion response from islets isolated from human non-diabetic donors shown here. However, the addition of mefloquine had no effect on islets isolated from diabetic NOD mice, where beta to delta cell gap junctional coupling is already severed due to the loss of the beta cells, and reached hormone secretion levels that were actually comparable to that produced by the young non-diabetic NOD islets treated with the connexin 36 blocker mefloquine, as shown on the previous slide. So we next tested whether we would see similar observations by trying to mimic the in vivo autoimmune attack in vitro by treating isolated non-diabetic C57 Black 6 islets with a pro-inflammatory cytokine cocktail for 24 hours, where we found that cytokine treatment also led to a significant reduction in islet GGAD2 expression encoding connexin 36, which has also been shown by other groups, which was associated with an increase in delta cell electrical activity at 1 millimolar glucose, here shown in red, when compared to delta cells from non-treated islets to the left in black, which remained electrically silent, which corresponded with a significant increase in somatostatin secretion and reduction in glucagon release in cytokine-treated islets when compared to the non-treated islets to the left. And we also saw a similar hormone secretion response in islets isolated from human non-diabetic donors treated under similar conditions, where neither the addition of gap junction blockers, carbonexolone, or mefloquine had any additive effect on the increase in SMASTAN release or reduction in glucagon. So together, these data suggest that when the beta cells are present and normally repolarized at low glucose under non-diabetic conditions, that this repolarization is capable of spreading into the delta cells via the gap junctions, keeping the delta cells electrically silent, enabling normal alpha cell glucagon secretion. However, when this electrical coupling is severed, as the beta cells are gradually lost in type 1 diabetes, this alters the electrophysiological profile of the delta cells, which enable them to fire more action potentials and release more somatostatin under hypoglycemic concentrations, which in turn inhibits alpha cell glucagon release due to an elevation in paracrine suppression. So the question is then, how many beta cells need to be lost in order to achieve maximal beta to delta cell gap junctional uncoupling? Now, it would be really difficult to do this experiment in the lab, so we instead turned to mathematical modeling using 3D maps of published architectures of non-diabetic human islets, where we identified that more than 90% of the delta cells remained in contact with at least one beta cell until more than 75% of the beta cells have been removed, suggesting that more than 75% of beta cell loss may be required in order for maximal decoupling between the alpha and delta cells to occur. And on the flip side, this also suggests that the maintained presence of functional beta cells may impose a protective effect against hypoglycemia. We then finally asked, well, can we improve then this impaired glucagon response to hypoglycemia from diabetic islets by blocking the action of this elevated SMASAN secretion from the delta cells at low glucose? So to test this, and because SMASAN receptor 2 is the most functionally dominant SMASAN receptor expressed by alpha cells, we applied the SMASAN receptor 2 antagonist called CYM154806 at low glucose, which significantly improved glucagon secretion from the diabetic NOD pancreas, shown here in red, to levels that were comparable to that produced by the adult prediabetic control pancreas in gray. And it also improved the impaired glucagon response from NOD-diabetic mice in vivo during an IP insulin tolerance test, when the antagonist was injected 15 minutes prior to the injection of insulin at time 0, where diabetic mice injected with the antagonist are represented by the solid red line when compared to vehicle-administered controls represented by the dashed red line, and pre-diabetic controls injected with and without the antagonist are represented by the gray lines. But what about human type 1 diabetic islets? So when applying the SMASAN receptor 2 antagonist at low glucose, this also led to a significant improvement in glucagon secretion from all type 1 diabetic donor islets in static incubation. So to summarize, the islet alpha cells from both non-diabetic mice and human type 1 diabetic donors show impaired counterregulatory glucagon secretion in response to hypoglycemia, which is also caused by an elevation in alpha cell paracrine suppression due to an increase in SMASAN release from the neighboring delta cells. Now, we believe this increase in SMASAN release is caused by electrical gap junctional decoupling between the beta and the delta cells, as the beta cells become more dysfunctional and are gradually lost with disease progression. And then could this explain the abundant clinical evidence mentioned earlier supporting the presence of residual beta cell function and having a protective effect in reducing the risk of episodes of hypoglycemia and severe hypoglycemia in individuals with type 1 diabetes? We believe it does, and that the simple answer to this phenomenon could be that through electrical coupling, the beta cells serve as an electric brake on the delta cells, preventing them from releasing too much somatostatin at low glucose. And finally, this impaired glucagon response to hypoglycemia seen from both non-diabetic mice and human type 1 diabetic donor islets could be significantly improved by blocking this increased SMASAN signaling on alpha cells by applying a SMASAN receptor 2 antagonist, supporting the use for SMASAN receptor 2 antagonists as a potential therapeutic for preventing the risk of severe hypoglycemia in individuals with type 1 diabetes. And just to add, a Canadian-based company called Zuccara Therapeutics, who are testing their own SMASAN receptor 2 antagonist called ZT01 on its ability to improve the impaired glucagon response to hypoglycemia in individuals with type 1, shared some of their phase 1A findings 2 years ago at the ADA and EASD, where they reported that 90% of type 1 subjects who underwent a hyperinsulinemic hypoglycemic clamp injected with the SMASAN receptor 2 antagonist did have an improved glucagon response to hypoglycemia when compared to placebo controls. And I believe they're now undergoing a phase 2 study investigating the effects of ZT01 on its ability to prevent nocturnal hypoglycemia. And I'd like to thank my colleagues based at both Octem and the University of Gothenburg labs, and to our collaborators. A massive thanks to our leader, Professor Patrick Boersman, for his fantastic mentorship and support at both Octem and Gothenburg. And a big thanks to the islet isolation facilities, to Professor Patrick McDonald at the ADI Islet Core, Nordic Islet Network for Clinical Transplantation, and Islet Isolation Facility out at Oxford, helping massively in the efforts to strive research on human type 1 diabetic islets forward. Thank you to our funders and the reviewers on the paper, and of course, a huge thanks to the families of the organ donors. And just to add, if anyone is interested in the models presented today or the techniques, please do get in touch. So we've got the optogenetic mice, halorhodopsin in beta cells, and channel rhodopsin and halorhodopsin in delta cells in Gothenburg. So please get in touch with Hai Kiang and Caroline. And also, if you're interested in doing calcium measurements and GCAM animals specifically expressed in the delta or alpha cells, we have the delta cell GCAM mice now, as mentioned, but we're also getting the alpha GCAM mice really soon, or to contact our supervisor, Patrick. No, thank you so much.
Thank you so much, Tom. This was great. Really great work. Amazing. And I love the fact that you have such an excellent collaboration across borders with these other groups to kind of pull this across the finish line. I just had a couple of questions. I want to open it up to questions. Please feel free to drop a question in the chat, or you can raise your hand or unmute yourself. Feel free. In the meantime, I had a couple questions. So what are the off-target effects of this SSRT-ZA drug? Are there any, or is it a safe drug?
Yeah, that's a really good question because it's not just expressed in the pancreatic islets, it's
Right.
expressed in the brain, in the
Mm.
hypothalamus, in the cerebral cortex. It's expressed in the GI tract and the adrenal glands. Now, actually, somatostatin receptor 2 antagonists, they're currently being used as a diagnostic tool in positron emission tomography to identify neuroendocrine endocrine tumors, which also predominantly express somatostatin receptor 2. And they've been noted to be well tolerated. And as mentioned, the Canadian-based company Zuccara Therapeutics testing their antagonists, which it has showing promising results, improving counter-regulatory glucagon secretion in response to hypoglycemia in individuals with type 1. They've reported no major adverse effects so far. Now, what the chronic implications of taking somatostatin receptor 2 antagonists are in terms of off-target effects, I don't really know, and I think that information will be much clearer with the ongoing clinical trials.
Yeah, that's really very cool. We'll keep an eye on them. If we wanted just to back up even behind some of the behaviors of these cells, right? So there's an impact of the vagus nerve on, at the beginning of all of these, somatostatin release, correct? So for instance, the vagus nerve is releasing acetylcholine that binds to the M3 receptors on the delta cells, and then there's a release of somatostatin. How does that play into your model? Is that like a backup, or is that the primary, or is it like um, a co-regulator?
Yeah, so I mean, it may play a role, of course, in vivo, but the fact that we see elevated somatostatin in isolated islets where innervation is severed
Mm-hmm.
suggests it really it is an intra-islet or is partly driven by an intra-islet defect. So this is the surprising thing. Now,
Yeah, I know.
Now, what's driving the increase in intra-islet somatostatin? Well, decoupling is possibly one mechanism, but there may be other factors as well. And for instance, it's been reported that the alpha cells may actually begin to produce GLP-1.
Mhm.
And we know that this happens in type 2 diabetic alpha cells. But it's now been reported that there is an increase in PCSK1 expression in recent onset in the islets of recent onset type 1 diabetic donors, and PCSK1 encoding prohormone convertase 1 will process pro-glucagon to GLP-1 or glucagon. GLP-1, of course, will also stimulate somatostatin release at low glucose. So this may be another mechanism, and this is something we're looking into in great detail at the moment.
Yeah, parallel backup.
Mm-hmm.
So that's very interesting. So then I did have to touch on sort of one of my favorite topics is the primary cilia on the beta cell. Michele Solomena and others just had a really great paper in EMBO in March. Somatostatin triggers local CAMP and calcium signaling in primary cilia to modulate pancreatic beta cell function. And they did look in type 2 donors in islets, and they found that the primary cilia length was reduced. And it was associated with a reduction in interactions between the delta cells and the cilia. So that's really interesting, isn't it?
Yeah, I'm afraid we haven't looked at the primary cilia, but this is something we'd be interested in looking at for sure.
Yeah, I mean, they
Mhm.
also found like the islet delta cells are positioned very near, and they release the somatostatin onto the primary cilia of other islet cells, including beta cells. So it's kind of curious that, that's like another way in, basically, for the somatostatin impact.
Yeah, absolutely. Yeah.
Well, there's more to do, obviously, after all this discussion, and it just, it makes sense that there are multiple systems controlling such an important islet organization, and especially in humans. And it will be up to you all to dissect how they work in parallel or at different times, or however it is they're working. So thank you again very much, Tom. You really, this is a great talk, and it's really interesting work, and it's fabulous that you are open to collaboration and sharing your, your resources and tools. So thanks again, and I hope you have a great rest of your day.
Thank you.
Thanks again. Bye-bye.